All right. Welcome, everyone. Good evening, and welcome to this webinar tonight. On ongoing research in myeloma. So, more specifically, developing less invasive detection methods using blood-based cell-free DNA tests. Very exciting topic. I present myself, Karine Gravel. I work for Myeloma Canada. I'm a regional manager in taking care of all the education material with Myeloma Canada, but also responsible for community engagement. I have here Chantel Wicks, my colleague, and I will let you introduce yourself, Chantel.
Hi everyone, I'm Chantel. I also work for Myeloma Canada. I am the Regional Manager of Community Engagement and Development. So if you are in the provinces of BC all the way to Ontario, including the territories of Nunavut, as well as Newfoundland and Labrador, should you need anything, I am the person to contact, and Kitten will be showing our emails as well shortly. So take out your camera phone, take a picture so you have our emails kept safe.
Wonderful. Thank you, Chantel. So we are very excited and honored to have Dory Abelman with us tonight. He's a PhD candidate in the Department of Medical Biophysics at the University of Toronto. Welcome, Dory. Thank you so much for being here.
Thank
So
you.
before we move on, I will take just a few minutes to share my screen and show you a bit of instructions for tonight. On So I would like to take just one moment to thank our sponsors. Of course, without them, we would not be able to do such educational material for our community of patients. Thank you so much. So as you can see, you have three panelists on your screen. Unfortunately, there is no way that we can see all participants and the audio is not activated. The way we will interact with you today will be using the Q&A function. So if ever you're on a tablet or on a phone, you just sometimes have to swipe on the side to get to the menu bar that you see on the screen. Please feel free to enter your questions throughout the presentation so that you don't forget. And at the very end, Chantel and I will be happy to coordinate the questions with Dory. Where can you find this video after? If you go on myeloma.ca, under the recently diagnosed tab, you will see there is access to the resource library where we have all the information, all the educational material, info guides, info sheets, but also access. If you go down, you'll see additional resources, and then you have access to our YouTube channel. Click on it and you'll get directly to our YouTube page. You can look for us on YouTube directly if you don't want to go through this process. And you type in Myeloma Canada, and if you see our gray and red flower, you're at the right spot. And then you can toggle between videos or playlists and have access to all our past webinars and some info videos. And if you hit subscribe, you'll be notified by email every time we add something. Just 1 minute to remind you that we have an app. It's called MC Lounge. This is actually a numeric platform, a bit like Facebook, to exchange with other people affected by myeloma, but very specific to our community. So no noise, no publicity at all. And you get to see members on a map that are around you, so you can maybe grab a coffee and discuss. Also, a quick link to subscribe to our newsletter. So the newsletter, if you're not part of it yet, basically you will receive from Myeloma Canada an email once a month to actually get access to our all the latest news about myeloma, treatments, research, community events, so on and so forth. As we discussed at the very beginning, so I'm Karen Gravel. I'm in charge actually of the provinces of Quebec, New Brunswick, Nova Scotia, and PEI. You have access here to my email if you want some help on resources, but also support groups, anything related to that. And these are the contacts for Chantal with all the provinces that she mentioned at the very beginning. So I'm going to stop there because I'm sure this is not what you would like to see today. So I will let Dory present the topic for tonight. Welcome, Dory.
All right. Well, hi everyone, and thank you very much for joining the webinar today. My name is Dory Abelman, and I'm a PhD candidate at the University of Toronto, and I work at the Princess Margaret Cancer Centre in Toronto. And I'm excited to talk to you today about an introduction to me, what I do as a PhD student, and the kind of research that I engage in, looking into some of the projects that I'm working on monitoring myeloma with cell-free DNA, developing new ways to detect minimal residual disease in myeloma. We'll then talk about some other projects if there's time and have a bit of a Q&A. Throughout this presentation, I have some interactive components with a tool called Slido. The way that this works is you go to this website, slido.com, and you type in myeloma, or you scan this little box with your phone. Alternatively, you can write in the chat. My first question before we get into this workshop is how would you rate your familiarity with multiple myeloma research? So I'm going to leave this open for about a minute, and you'll be able to see your responses live on the screen. All right, thank you to everyone who's voting. So we have a split between very familiar, somewhat familiar. Looks like most of the group is somewhat familiar. So what I'm going to do then is I'm going to speak a bit about what my role as a researcher looks like, and then give a little bit more context into some of the research that we're doing. Thank you very much to everyone who casted their vote. If you didn't put in a vote yet, no worries. Um, you'll have other opportunities to engage with interactive polling in later parts of this presentation. So to start about myself, so my name is Dory, and I'm a PhD candidate. I completed my Bachelor of Science at Western University, and then I joined the University of Toronto as a Master's of Science student. I since then passed a qualification exam and became a full PhD student. What does it mean to do a PhD, and what is being in a PhD program actually like? Well, typically the program spans anywhere from 4 to 7 years, and our goal in the program is about creating new knowledge. So really putting forth the cutting edge, advancing new research, and coming up with ideas that haven't been fully developed before. So what it looks like is conducting different experiments, analyzing data, reading a lot of scientific papers to understand where the field is, and trying to push the boundaries of what's known with our experiments. I put this graphic that I found online because I thought it was funny and quite relevant. Typically, we go into this program, we say, everything's going to be so smooth. I'm going to work really hard and I'm going to discover this amazing thing. In reality, it's actually quite difficult. So we run into a lot of challenges, things that we didn't expect to encounter. We encounter things that we don't think will work, end up working, and things that we think will work don't end up working. But overall, it's a fun adventure where I learn a lot. And I hope that in the work that we're doing, some of which I'm going to share today, will help push the whole field forward. What do I actually do? I think there's a bit of a misconception that all people in research and in science wear lab coats and work in a lab all day. So actually, when I was doing my bachelor's, I did work in a lab in my bachelor's. research was all with cells, with the lab coats, with the microscopes. But currently, my my work looks like this picture over here. This is actually a picture of my desk, which I took today. And you can see what's on the desk is a lot of code. And the reason that I do so much coding is we work primarily with DNA, which is so small that I wouldn't be able to see it even with a microscope. So we have computers give us the code of the DNA, and then I use computers to analyze that code and learn from that code. So I really do spend most of my day writing code, and this is by looking at the samples which we get, which I'm going to talk about how those samples get to us and what we do with them. And the goal of this is to better understand why drugs are succeeding or failing and discover new ways to monitor myeloma, and then share those findings with the rest of the scientific and medical community so that they can one day be integrated into patient care. So where does this research start before we even get to samples? Typically, we have scientific articles, scientific publications. That's like the current state of the field, and somebody, a clinician or a scientist, will get an idea from this existing research, which will then inspire them to create a set of experiments or a study to see if that idea is actually true. For example, can we use DNA from blood, from regular, like peripheral blood, to monitor myeloma? They'll then apply to a funding agency. In this case, I put an example of the federal government, the Canadian Institute of Health Research, which provides a lot of resources for researchers to carry out their research. And if it gets funded, they'll submit an REB, so a research ethics board, which will then have all these components, and they'll have to prove to the research ethics board that of a hospital or an academic center that the experiments they want to do, if they involve patients or animals, that they're safe, that they are worth any kind of risk that may occur to make basically the maximum benefit possible to patients. And the Research Ethics Board is all about protecting patients and ensuring that we have again the most benefit with the least harm. And once that board, if they then approve that study, physicians can then request patients to collect samples. I work with a lot of peripheral blood samples, so that's like a regular blood draw, and bone marrow aspirates or bone marrow samples, which are also withdrawn in the clinic. Those samples are then put into tubes. Some of them stay at the hospital where they were extracted and used for testing there. But some of them, if a patient has consented to be a part of a study, will get into a truck. I mean, typically we work a lot with FedEx, but also with other companies too, and get shipped over to us. Once we get the samples, we get them in tubes of blood, typically. If it's blood or if it's bone marrow, we'll get like a tube of bone marrow cells. We'll then take the DNA or extract the DNA out of those samples using different kits that we have in the lab. So this is a picture of our lab. And what we can see here are pipettes. Can you see my mouse? Yeah. So these pipettes, they're kind of like little measuring devices, and I can take a very small amount of substance very precisely from those samples. And then once we have the DNA, we'll typically store it. So here we have a freezer, a -80 freezer full of thousands of samples for different projects. This freezer has samples from a bunch of different labs, and they go in these little boxes. And when we keep them cold, they can stay they can stay fresh for a long time. And I mean, we can use them in the future, many years from now. If somebody has an idea later, for example, that's if a patient has consented to be a part of a biobank. So biobank stores samples for a long time. The samples that I work with are mostly for specific studies that patients have consented to be a part of. So they'll be in this fridge. Um, when we're ready, so typically when we have a group of samples that are all ready, we'll put them on a fancy DNA sequencer machine. So on the left here is one of the new machines that we have in the lab. This is actually across the hall from where my desk is. I'm not the one who's actually extracting the samples most of the time and putting them on the DNA machine. That's done by research technicians and other lab members who are really excellent in specializing in that area of research. This machine, it's about the size of a fridge, maybe a little bit smaller. And what we do is we put the sample, the DNA inside, a small amount of it, and it will use this fancy chemistry to go and tell us what the code is of the DNA. So why is this code important? Because in myeloma, changes to DNA are what cause the changes to the cells that we end up seeing. So here I have a schematic of a bone marrow, and I'm showing a healthy plasma cell over here. But then if we have damage to the cells in the bone marrow, we can end up with cancerous cells. And this is a myeloma cell. And we know what these changes are generally. So here's an example from a research article showing that cell. And these are the different stages of myeloma from MGUS, monoclonal gammopathy of undetermined significance, through smoldering multiple myeloma, active multiple myeloma, and extramedullary disease. There are different genetic changes which occur at each of these steps which cause the cell to change, as in this example. And if we can find those changes, then we can more accurately diagnose a patient with myeloma and be able to understand what drug would be best for them to use and help them manage their risk. One more thing to mention about this is the changes are quite different between people and even within the same tumor among people. And this is called heterogeneity. And here I have a little example. So we have cells in a tumor over here that all look very different from each other. And then we can have changes in the DNA, and we can have changes to all these other factors. So we're not just looking for a specific change in every patient, we're actually looking for different changes in each patient to help us better understand what the best way to treat them would be. Now, the traditional way that we look for these changes is through bone marrow aspirates. And here's shown an example over here with that aspirate being taken. So we have a big needle which goes into the pelvis and it captures some bone marrow, which gets sucked into the tube. And then from that needle, we can then get the cells and do things like flow cytometry. We can do things like FISH or fluorescent in situ hybridization, which is a way of knowing what the changes to the DNA are, or we can do more advanced DNA sequencing, like I showed in that other slide. Now, another way that we can monitor myeloma, other than this bone marrow, is through looking at things like the M protein. Now, M protein we can look at in the blood, for example, with things like immunofixation. But some challenges with these technologies currently is the bone marrow aspirates are highly invasive, which means they're not very comfortable for people to get. It can be painful. And another limitation is when sometimes we're only capturing cells from one region in the bone. Um, and myeloma can have many different places in the bone where we have a tumor. So one other concern here is that there are cells that are outside of the region of this area that's being sampled, and they're missed. So we call this patient, for example, negative if we're monitoring them after their treatment, but they were just cells here which we didn't get. So there's one limitation. And then a limitation with the M protein method is currently it's not super sensitive. So we can use it around the time of diagnosis, but we're hoping to get even more sensitive for the MRD time points. And there are some methods to improve the sensitivity of M protein, which I'll talk about. And what does the schematic over here show on the right? So this is kind of a cartoon where we can see different proteins in the blood sorted by their mass or how heavy they are. And typically we see something like the blue curve over here. But in myeloma, we'll get a big spike of other proteins, which are kind of junk proteins. They're antibodies, which aren't used by the body in a productive way. And they're produced by all of those plasma cells, which are just plasma cells typically produce antibodies. And because there's so many of them now, we get that big chunk of antibodies, and that's called the M spike. So how can we improve this? So our lab is looking at using cell-free DNA. Now, what is cell-free DNA? Well, cell-free DNA, so you can think of cell. In the bone marrow, we're typically pulling up cells. Cell-free DNA is DNA that's outside of those cells. And this is a schematic showing how it's released into the blood. So we have all these cells in our body, and through different ways, for example, like when the cells die, they release their DNA into the bloodstream. And then if we draw a person's blood, we can then get that in the tube. Now, in cancer, the tumor is also releasing DNA, those plasma cells, and our goal is to be able to identify those tumor DNA pieces in the blood as well. And this whole field in general is called liquid biopsy because it's like instead of getting the DNA from the cells directly in the bone marrow, we can get pieces of DNA that broke off from the bone marrow and are now floating around in the blood through a regular blood draw. What's really exciting about DNA is it's really a one-stop shop for lots of different information that we can learn about regarding the development of myeloma. So here I'm showing different samples that we work with in our lab. So we have blood over here at the top, and its different components, and then bone marrow, which we separate into the cells inside the bone marrow and the liquid around those cells. And we put it onto that fridge-like machine, and we can look at things like copy number profiling. That's how much DNA is there. Do we see a lot more DNA in chromosome 1 at the end, for example? And that would be an amplification in 1q, which would affect the way that we treat a patient. We can look at mutations, which are changes in the DNA. For example, in a place where we'd expect a G, we see a C instead. What that can do is change the way that a cell behaves. And we can also look at immunoglobulins, which help us understand if a myeloma cell clone is present, which I'm going to show examples of in the next couple slides. So let's start first by actually looking at DNA and what that looks like. So what I'm showing you over here is a bunch of different bars. This is what we typically see on our end from the machine. And what we can see is, so each bar is like a piece of DNA, which a machine scanned essentially. And then I use software to put all the DNA pieces together um, where they belong in the genome. So we have a certain order to the DNA in our bodies. And by looking at the order on the pieces of DNA that the machine scans, we can know at what part of the body it belongs to. So the gray means that it's in a place where it belongs. The blue over here with these different colors means that it seems to be belonging in another place. So what's really interesting is here we're looking at chromosome 11. So this is one of the chromosomes in the body. But all these blue and green over here map to chromosome 14. So what we just picked up in this patient is T1114, or a translocation between chromosome 11 and 14, which means the DNA broke and rejoined. And why would the DNA do this? It's because we can have these very, very strong enhancers or things that cause the DNA to be very, very expressed, meaning very active. And then we can have these genes that cause cells to grow very aggressively or very fast. And what we have in this case, we have a very strong enhancer on chromosome 14, IgH, immunoglobulin heavy, bind to this gene called MyoV, which is myeloma overexpressed gene 1, which causes cells to grow really crazily. And what we identified in this patient is that this DNA, that this gene, which would cause the cell to grow very fast and divide very quickly, is being very active. So this would not be good generally. But because we know that it's there now, we can choose how to treat a patient appropriately to account for that. But the most interesting thing here is everything that I'm showing you is from cell-free DNA. So this patient, we didn't have bone marrow for them because there wasn't enough cells in their bone marrow for us to use. This is based just on the DNA that we took out of the peripheral blood. And what we see is this patient is here at baseline. So when they started the study before they started a new treatment, we can see that they have this translocation. And at the top, we see the same patient a few months later after they got some treatment. And what's interesting is that patient only had a partial response, which means that they didn't respond very well to that treatment. And we can still see this piece of tumor DNA in the blood. So when we don't see this in the blood, generally then that's a good sign. It means that there's very little tumor in the blood, which typically means that the actual size of the tumor in the bone marrow is smaller. So what other things can we see from the DNA in the blood? Well, another thing that we can do is we can look at mutations. So these are changes in the DNA. Here I'm looking at a gene called NRAS, which is on chromosome 1. It's a gene that when it's changed in this way specifically, when we get this G, where we typically don't have a G in that location, the cell can divide much quicker, which is a common feature in cancer, which leads to worse cancer outcomes. And what's interesting is we can see this big G here, which means that we were able to detect this cancer mutation using only DNA from the blood. Now, this is going to be really helpful for deciding how we're going to treat the patient. And again, for this patient, we didn't have bone marrow available. The other thing that we can do is we can look for how much DNA there is, so the copy number. So the way that we read these plots is each number represents a chromosome. So we have 22 autosomal chromosomes in the body. That's 1 through 22. And then we have the sex chromosomes, X, 2 copies of X for a female, and 1 copy of X, 1 copy of Y for a male. But what I'm interested here is this end of chromosome 1, chromosome 1q, because if we see this in a patient, that typically means that the patient is higher risk, so will need to be treated in a slightly different way. Now, we didn't have the bone marrow DNA from this patient, so again, using only the blood, we were able to see this 1q. Now, again, after a few months of treatment, the patient didn't respond so well to the drug, and still we can see the 1q. In patients that respond very well, we typically don't see that 1q anymore. So this is another way that we're using DNA in the blood to be able to monitor patients. Another way that I want to show you is by looking at the immunoglobulins. So in myeloma, we typically have a lot of cells which are a clone of one cell, which means that they're copies of that same one cell, which kept dividing and dividing like crazy, and to the point that it took up all of the space in the bone marrow, and there wasn't space anymore for the bone marrow to be able to do the things that it normally does to sustain us and make us strong. So one of the ways that we can detect if that patient has myeloma is by just seeing if out of all the immunoglobulins that we see, so these are little receptors on the cells, if a lot of them map to only one clone. So on the right over here, I'm showing you an example of healthy cells. And what we see here is there's a whole bunch of different clones that we see. So each each kind of like rectangle in the bar is a clone. But on the left, I'm showing you that out of all the clones, you can see the gray one takes up over 95%. So that means that out of all of the plasma cells that we're seeing, 95% of them map to a single clone. So this we'd only see in myeloma. And this we found only again using the blood. And what's interesting again is after treatment, we still see that really big clone, which is indicative that the patient it still has some tumor present because we can see it in the blood. So here's looking at that at the same time for another patient. So we can see they have this clone over here, which persists at the beginning and through their first treatment time point. We can still see here this patient had a 1Q. All of this is using only the blood. We didn't have the bone marrow for this patient. Then the 1Q gets smaller, and then eventually we don't see that 1Q anymore. And this patient had very good partial response. Also, we no longer see that gray tumor clone, which we saw at the beginning. So these are two ways that we're using DNA from the blood to monitor myeloma. I next want to show you a comparison of mutations. So I showed you before an example of that single NRAS. Um, what I'm showing you here is these are different mutations that we found in the bone marrow. That's shown on the left, and each dot is a mutation. And on the bottom, we have two different kinds of cell-free DNA. So the first is from the peripheral blood, and the one over here on the left is from bone marrow. This is the liquid that's surrounding the cells in the bone marrow. And what we see again here is we're able to catch all of these mutations in the blood, and in the bone marrow, we only missed this one here, this TRAF3. So this is showing you across many patients that we're able to detect the same mutations, which are important for monitoring the disease in the blood that we see in the bone marrow. So what does this all mean for for those listening? So what we're doing is we're working to improve the detection of genetic alterations that we would typically need the bone marrow for using cell-free DNA. And the goal of this is to provide patients with options to find their most important features, genetic features, such as those translocations, mutations, and copy number changes that we looked at without using baseline bone marrow samples in cases where those aren't available, or to provide patients with options to use this cell-free DNA to find features in cases where they maybe want to do it more often. Because of bone marrow, we can only do, for example, every couple of months because it's such a big procedure, but blood draws are easier. So we're hoping, which what I'm going to show you next, is how we use this to track patients over time. So what I have over here is a myeloma patient and with lots of treatment time points over here. So as we go across, the patient gets a bone marrow transplant, they have post-induction transplant, and 2.5 years of maintenance. And what we can see is this big pink purple clone, this immunoglobulin, which we saw at the beginning, we still see over all of these time points. Now, what's interesting is this patient was actually negative by flow cytometry or multiparameter flow cytometry right over here at this point, which I'm highlighting with my mouse. Number 5. But again, we still saw this clone and it still persisted all the way through. So these are other ways which we can track a patient. So now we saw these immunoglobulins, we saw these mutations. All of these focus on a kind of sequencing called targeted panel sequencing. That's when we look at a few key areas and we look at them very, very closely. What I'm going to show you next is a new approach that we're trying out, which we think will help us find disease in um, even lower disease burden cases. Let's go to cell-free DNA for MRD detection. By the way, this picture here is of someone actually tracking a financial market, so you can see when people bought and sold. But I put it here because I like it because it shows the flow of how something changes over time, which is what we're trying to accomplish. So why does early detection matter? So this idea is when we have myeloma or any cancer, it starts with one cell, which has those genetic changes. And over time, that cell grows and grows until it becomes many, many cells. And then if those cells start to split off and go to different places, we call that metastasis, which can be very difficult to target because now we have cells in all these places in the body. When I'm talking about minimal residual disease or MRD, I'm talking about once we treat the patient being able to see when they have a very, very small number of cancer cells. So maybe less than 1 in 1 million healthy cells in the bone marrow. And the reason that this is really important is because the more we know how much cells they have, the more precise we can be, the better that we can predict if someone's going to relapse and when. So what this plot is showing you here is different ways that we score patients. So for example, VGPR, CR, stringent CR fall all around here. But even if someone has stringent CR, they can still progress after, let's say, a couple of years. When we hit the MRD time points, we want to be able to say that this patient has maybe even 10 or less cells. And if we really can't detect those cells at all, then we know that that patient is likely to be myeloma free for a very long time. Now, there are currently different trials in place trying to see if we detect that the patient has a higher number of cells, if we start treating them early or changing their maintenance, will that bring the count even lower and keep them negative for a very long time? But for those trials to work, we want to be able to tell very, very confidently to a super low level how many cells are present, which is what I'm going to present to you next. So our approach is called cell-free DNA whole genome sequencing. So what do I mean by whole genome? I mean looking at all of the chromosomes in the body. So we have 3 billion base pairs approximately in our DNA. And traditionally, we had to look at only a small amount of them. So, for example, those plots that I showed you earlier with those mutations and those immunoglobulins, we're only looking at maybe 100,000 out of 3 billion because it was just so expensive to look at all of them. But now sequencing technology, which is the way of getting that information, has become a lot less expensive. And also, we realize that we don't need to go that deep to find it. So what do I mean by deep? So what I'm showing you here is the probability or how likely we are to find a single needle. So imagine you have a needle in a big haystack. This is the probability of finding the needle in the haystack if you only have one needle. And depth, in this case 10,000, means that we look at each part of the genome 10,000 times. This is like taking a lot out of the pile. So 10,000 means if you took out 10,000 pieces of hay from this pile, how likely would you be to find this needle? If there was one needle in 100,000 pieces of hay, well, you'd be very, very unlikely because there's only 1 in 100,000 and you only took out 10,000. So very unlikely. But what if you have 10 needles and the you had 10 needles per 100,000 and you pulled out 10,000 pieces? So now you're actually much more likely, but still quite low. You can still miss it. What if you had 10,000 needles per 100,000 needles and you only pulled out 30 pieces of hay? Your chances of finding any one of those is actually much higher now. Um, And another way to think about this is related to the myeloma cells. So when we're talking about minimal residual disease, instead of one needle in 100,000 pieces of hay, it's one cell, one myeloma cell in 100,000 healthy cells. And what are those needles? Those needles are things that we're looking for. In this case, changes in the DNA or variants. And what that looks like is this. So we've developed a strategy where we look across the entire DNA and we find the whole genome, all of the places where the cancer, the myeloma, has made changes in the DNA. And because the myeloma cell is dividing so fast and growing so fast, there are actually many places in the DNA where we find changes. And on average, we find about 10,000 changes across the whole DNA per cell. And what that looks like is here. So at the bottom, we have the different letters, and you can see there are all these changes here. But if we only looked at a small number of the region, we would actually miss those areas. So what we do is we take the tumor and we also take the blood and we sequence that in the fridge machine. We look for the changes and then we see if we can find them over time as the patient has post-induction therapy, transplant, and maintenance. So here's an example of what that looks like. So we had a patient which had a mutation in TP53. So that's one of the genes that affects the way that cells divide. So when we have this, it can cause the cells to grow very fast, which is not good. So knowing that we can affect the way we treat. And we only saw that one time in that patient when we looked across the whole genome because we looked only 40 times there instead of 5,000 times. But we didn't just find one mutation. We had about 10,000 that we were looking for. So we're looking for 10,000 different features now. And if we can find just a small number of them, then we can confidently call that patient as positive or negative. And here's what that looks like. So when the patient started, we had at diagnosis, we had 10,000 mutations that we found in their bone marrow. We then found around 2,000 of them in their blood. The patient then had induction therapy, achieved a CR, a complete response, had a transplant, and then was negative by clonoSEQ, which is from Adaptive Biotechnologies. It's a kind of MRD test that's performed on bone marrow. But what's interesting is at the time when the patient was negative by clonoSEQ, we still had 477 out of the original 2,000, which we found in that patient at that time point. So we had evidence here that the disease wasn't fully cleared. A few months after that, we now see 2,486 changes. So there's even more and more changes that we're seeing. And then 2 months after that, the patient relapsed. So this is a case of how our technology can help us find if a patient will relapse in a time where it was even negative by a test like clonoSEQ. And why do we think this is because the bone marrow-based tests are only looking at a very small part, again, of the bone marrow, just the region that are being observed by the bone marrow biopsy. So what we think what happened in this case is that the patient, so again, here's an example. We think that the patient's myeloma cells were not in this area. They were maybe back here or maybe here. Um, but all of these cells are releasing DNA into the blood. So in the blood, we were able to see from all the cells instead of just the area where the needle was. Another thing that's interesting, which we can do with this, is because we're looking at, again, the whole genome, so all of the regions of DNA, we can also see other things like changes in the amount of DNA in specific places or the copy number. So what we see here is that same patient that I just showed you the curve that went down and then back up again. Here we see how much DNA they have from chromosome 1, 2, 3, 4, all the way to 22. And what we see here is the patient had a big gain here in chromosome 3, and also in some other chromosomes. This is what we'd call hyperdiploidy, which actually is favorable prognosis in myeloma. This is what we got from the bone marrow. But what's interesting is when we look at the blood, we're able to see those same areas. So it's, for example, where there was a gain in the bone marrow, we also see a gain in the blood. Now, if I fast forward to 2 months before this patient relapsed, all of a sudden we start to see these strange things. So the gain in chromosome 9 stayed the same, but all of a sudden we start to see a loss in chromosome 4. So if we look closely, we never saw this loss before. So blue means that there's normal copies, two copies, one from each parent. And then in cancer, we see changes like losses or gains. So the reds are when they gained copies and the greens are when they lost copies. So what this indicates to me is that the cancer actually evolves. And the clone, so the part of the cells that changed, what happened was the treatment killed most of the cells, but they were a small number of cells that survived the treatment, and they didn't have as much competition for food or resources anymore because the rest of those cells were killed, and they were able to grow quickly. And when that patient relapsed, they relapsed with the new clone. So part of the reason also why a patient might be negative, if a test is only looking for a specific cell clone, but the cells changed at relapse, a test may not be able to pick that up um, as as well, although there are ways to work around this. So this is another interesting thing in our technology. We can look at how the cancer evolves. And if we know, so this is 2 months before the patient relapse, if we know already that we're starting to see changes, there are new clinical trials which are establishing if changing the way that we treat at this time can help stop the relapse from happening. And again, all of this here was done just with the peripheral blood. So no bone marrow. I'm now going to show you some other cases of patients which then relapsed and their standard MRD tests. I want you to focus here on the top bar. So these are 4 patients that we have, and we can see that as these patients were treated, they went down, they went down, and then they went up again, and then relapsed. Now, some of these patients were negative. So we had a patient over here, which was negative by flow, and then relapsed. Um, but what's interesting is we're always detecting some of these original tumor variants using our method. So the top here is when I used the mutations in the bone marrow, those changes, and we're looking for them in the blood. And the bottom is when I didn't use the tumor at all. We actually just looked at the blood at the beginning, found some changes in the blood. And what we see is they're actually highly correlated. So for example, when this goes down, the top goes down. So what I'm hoping from this is that in cases where we don't have a bone marrow available at diagnosis or before treatment, we can still do something with the blood for these patients. Now, another thing is if I look at how much this line changes, we can see in all 3 patients where we had um, like multiple time points after the first treatment time point, we have a big increase. For this patient, even a full year before, we can see that the patients are starting to increase before they relapse. So we're hoping that by seeing that a patient is increasing early, we'll be able to also help inform that patient if they should maybe change treatment, etc. So what we're doing now, this is really just the development phase. So we're trying out this technology in a small group of patients. We're seeing if it works. And then if it works very well, it would be tried in a bigger group of patients. And then if it's been validated to work across many, many patients, it can then be offered in the future. So right now, what I'm showing you, we're still working out the quirks, but this is some preliminary data from our method. So to summarize, some advantages of our method and what we're proposing is using cell-free whole genome sequencing. It's useful for being able to detect disease outside of the bone marrow, which if we look at a single site biopsy, this is only looking at a small area, may miss. We're relying on blood, so this is less painful and less invasive than traditional bone marrow biopsies, allowing for more frequent and accessible collection. So what I'm hoping is if this ends up working in much bigger cohorts, that someone could just go and get their blood drawn maybe every 3 months at their local blood processing center, not having to go to a big hospital. And then that can be sent to the hospital or center to sequence to tell the patient if they need to come in and maybe go back on treatment. We're also able to capture genetic changes. So we looked at those copy number and the development of new clones. And we can also track how these mutations change over time to help us see if the patient is developing a resistant population of cells. Okay, so it's 7:22 now. So what I'm going to do is I'm going to wrap up. I have two questions that I want to ask you, the audience, and then we'll go into an open Q&A session with me. So the first question I want to ask you, so this is the same as the Slido that we had before, is if you could choose one area for future research in myeloma, what would it be? So is there anything that you heard that you thought was really interesting or that you want more focus on research to go? We have more sensible detection, CAR T, deep response data, and quality of life. We're also seeing in the chat stopping treatment periods. Okay, so there's a big interest in CAR T and how to attack residual disease. Okay, thank you to everyone who commented. The next one I want to ask you is one word to describe how you feel about the future of myeloma research? Hopeful, that's very nice to see. Revolution. Okay, a lot of participants are voting hopeful. So thank you for participating and promising. It's nice to see positive responses. Encouraging, hopeful, but guarded. Okay. All right, not fast enough. Okay. Well, thank you, everyone, for your answers. I really appreciate your time. I want to acknowledge everyone that I work with at the Princess Margaret Cancer Center, at the University of Toronto, and through these other studies where we get our samples from. There's a big collaboration, and I only work on a very small puzzle piece. And I also want to thank you and everyone who donates samples. None of this research would be possible without patients who are donating samples. And by donating, you're enabling researchers to really try to push the boundaries of what's possible and offer new services to patients. So I really appreciate your strength and resilience and commitment. And with that, I'd like to open the floor for any questions. So thank you so much for everyone for their participation. I hope you found this webinar meaningful, and I'm excited to answer any questions you may have.
Thank you so much, Dory. This is incredible information. I love the way you make your analogies and show pictures and images so we can easily understand the topic. And this is amazing. I mean, science is amazing. So very promising. I'm sure people are excited to see a less invasive test method, and hopefully this will minimize our use of bone marrow. So great. This is great. I think I'm going to open up to Chantal and see what we have so far in the Q&A section for questions to Dory. Just want to mention before that we had 4 minutes left. No worries. We're gonna take the time for you guys. The meeting will be extended by a few minutes so that we can answer questions. And if you have to leave, no worries. We will make this recording available to you on our YouTube channel in the next weeks. Chantal, to you.
Thank you, Dory. I echo Kedin's sentiments. Really interesting, especially I love the photos you included of the parts that we don't see, really. Your computer desk and the screens with all the stuff on there was already overwhelming enough for me, I will have to say. So one of the questions that we do have, actually, I'm going to do the first two questions that came in because I think they're a bit of a shorter answer. So the one was just to clarify, could this test be used in non-eligible transplant patients?
So we're still validating the test in different groups. So it hasn't been 100% validated, but I don't see any reason why non-transplant eligible patients wouldn't be able to participate. What we're doing is we're looking for changes across the DNA, and patients will have those changes whether or not they're eligible to receive a transplant, would be my guess. So I'd be very curious to give it a try and see if we can find those changes, but I don't see any reason why it wouldn't be possible.
Okay, and probably a similar answer in terms of needing to test further, but someone wondering if this type of test could detect AL amyloidosis.
So the way that the test is established currently, it only looks for general changes across the DNA, which we know that the patient had at baseline. So we wouldn't necessarily see the progression to AL amyloidosis, but other groups are looking at this from different cancers, and it seems to work quite broadly across different disease types. So I think in the future, with more fine tuning, it will be able to detect amyloidosis, but currently, this is something that I'll need to verify.
Okay. Um, a lot of questions here, so I'm just going to go through some of them. If you see them as well, feel free to answer. So we do have a question about just measuring blood tests. I believe you mentioned like 2 months uh, prior to a relapse, you noticed there was something changing in the blood. So if that relapse occurs rapidly, so when there's an M spike that climbs from, complete response over many months versus now. So how do we define what is a relapse? When you use this blood work to see a little bit of change, how would you answer that question?
That's an excellent question. So, um, that's actually something that we're actively trying to find out right now because even patients who will remain in remission for over 2 years, some of them will also increase by this method. So we're what we defined as relapse for the study was clinical progression. So, for example, more than 10% plasma cells in the bone marrow, the standard that our hospital uses. But sometimes we have patients that increase very highly, which don't relapse for some time. So this test is going to need a bit more calibration before we can confidently say that this patient is going to relapse, let's say, in X amount of time. But currently, we're able to detect residual myeloma changes in the DNA even a year or more prior relapse for the patients we've analyzed so far.
So I can tell by the questions coming in, our participants tonight are very excited about the possibility of this becoming a reality. And when I say this, I mean the blood-based testing to replace the bone marrow biopsy, and you could probably understand why. So one of the questions that we have, I think, is one of our members who's forward thinking about how is this going to turn out? Should this be proved super successful? So do the cancer centers have the equipment in their labs currently to do this type of test? I know you showed us a really interesting machine. So is that kind of in the cancer centers now, or are they going to require new equipment? And then if so, will your report or will there be further reports on looking at the cost benefit analysis to having a blood test versus a bone marrow biopsy? Big question, Dory.
Yeah, so most centers don't have these big DNA sequencing machines. I think the one that we have, we're one of two actually in Canada, which have well, now three in Canada that have the machine, although it's becoming much more common. A lot of centers had older DNA sequencing machines, which were very expensive. So part of the reason why we're able to do this is because we can use a much less expensive, newer one. So what I anticipate is that there'll be a few key centers. So for example, right now, the Ontario Institute for Cancer Research accepts referrals from different clinicians who will ask their patients to get, for example, a similar test. And then that blood sample will be shipped in the mail. So we can actually store them at room temperature for quite some time. So what I anticipate is other hospitals, other centers will order the test. The sample will then be shipped to a center that has the machine. It'll get sequenced there, and then we'll send the report back.
Okay, again, lots of questions. So I'm going through all of this. One question I think is short to answer. Um, if someone wants to donate their blood, Dory, for this research, can they go about doing that? Or because I feel like you'll have a lot of people knocking on your door if that's the case.
Oh, thank you. Well, you have to speak to your clinician about wanting to donate samples. So a clinician will inform you if you're eligible to participate in a trial. There are lots of trials going on, and they'll be able to enroll you if you're deemed eligible.
Okay. Um, Gosh, there's lots of fantastic questions, by the way. There was a question about for those on the call who have had the had more than one bone marrow, we shall say. So someone who has had more than one bone marrow biopsy, if that bone marrow biopsy is kind of taken from what seems to be the same place, the left side of the hip, is that standard procedure to have kind of the bone marrow biopsy taken from the same similar spot?
I can't answer that question, unfortunately, because I'm not the one doing the bone marrows.
True.
I just get the sample afterwards. So I'd recommend to speak to your clinician, about this.
Perfect. Um, does someone have to have active myeloma in order to take part in a test like this, or can it be done with someone who is stable?
The way that our test is organized now is we'll recruit patients when they have active myeloma. We'll make a barcode kind of what their myeloma looked like at the beginning, and then we'll look for that over time. So currently, we don't offer this to patients who didn't have a sample available at baseline. But I hope in the future, I actually skipped over this. We're looking at other ways to measure disease in the blood by looking at changes to the DNA which are completely independent of the tumor. And one of those is how long or short the DNA is. So what's interesting is the DNA in normal cell, like in healthy cells, it's protected by all of these proteins called histones, and it makes the DNA very stable. So when we see DNA from healthy cells in the blood, it's actually quite long. The cancer cells, they're growing so fast that their DNA um, is more open because it things have to go onto it to help the cell grow. And that DNA is very small and like unprotected. So one thing that we're looking at is if we can tell if a patient is MRD positive or negative based only on the length of their DNA. And in a small subset of patients, been able to see that MRD positive patients by flow cytometry consistently had shorter DNA than MRD negative patients. So in the future, we're hoping that there'll be ways to offer tests like this to patients with no tumor at all. This field is called fragmentomics, if you're interested for more information on this kind of technology.
Wonderful. How long does this take? How long does the analysis of a cell-free DNA take?
Okay, so currently in a research setting, it takes approximately maybe 3 weeks. However, in a clinical setting, it's possible to do it. I think the fastest it's ever been done was something like 4 days. So it can be done faster if it needs to be done faster. It just requires having more people on hand to process the data. But the way that we analyze it is very automated now, and we can often, in as little as 2 days, get results once we have the sample sequenced.
Okay. How far away do you think we are from this type of DNA sequencing being used on patients in the real world and not just in clinical trial? You might not know the answer to this story, but
Yeah, so I mean,
thought
currently,
I'd ask.
patients in Toronto at Princess Margaret can have their clinician order this test. Typically, they'd enroll them on a study, and then they can have the test done. Um, before it's more broadly available, I would think it's hard to give an exact time, but probably we're looking at something like at least 5 years, which is typically the time that it will take us to be validated on a very large cohort, because we don't want to be offering um, things unless we're absolutely certain that they work to patients. So um, depending on how successfully it works in more and more contexts, which is what we're looking at now, that will determine it. But I'd say at least approximately 5 years would be my best guess.
Okay. Yes, that seems to be an accurate answer. So we have a question, someone asking about beta lymphocytes, and do damaged beta lymphocytes die off? And if so, what is their replacement time?
So I don't work specifically with those damaged beta lymphocytes. So I can't give a specific answer right now. But I'm happy to follow up later and see if I know somebody who can help with that.
Okay, also have a question about whether or not this type of test could detect extramedullary tumors.
Yes, so that's actually one of the benefits of our test is we're able to detect tumors anywhere in the body as long as they have some sort of semblance to the original tumor. So we actually think in the cases where we called our test positive, but flow or clonal-seq was negative, that those were extramedullary cases. So we need some more verification, but we're confident that we are able to detect extramedullary disease.
Okay, we do have someone who's asked about something called OGM, optical genomic mapping, and wondering if you heard of that, if that's relevant, if you could explain a little bit more. If not, we understand that it's a separate thing. So,
Yeah, so optical gene mapping is another way of getting kind of the code of DNA. It basically has much longer strands of DNA, so it's actually very useful for looking at certain kinds of changes in the chromosome or like the bands of DNA. I don't work with it specifically. There are people in our center who do use it, but it's just another tool that someone can use. I think in our case, because we're most interested in specific mutations, using this the fridge kind of Illumina sequencing it would be more relevant to our case, but they are very good use cases for optical gene mapping as well.
okay, and we have time for one more question because there's quite a few, so we'll just go with one more. For those on the call who are out of province, say, they're not in Ontario, is it possible for them to speak to their doctor about having a blood sample for your test and research, or are we bound by provincial boundaries for this?
Yeah, so most of the samples which I've shown come from a study called the M4 study, which stands for Multiple Myeloma Molecular Monitoring, and we collect samples from 12 sites across Canada. So from all of the major um, myeloma hospitals across Canada. We get samples from most of them. So yeah, you're welcome to talk to your clinician. I'm not sure if the study is still recruiting, but there are always lots of studies coming up. And it's excellent to hear that you're interested. So thank you.
Yes, it is wonderful to hear when patients are like, absolutely, you can take my blood. You can use this. Like, because that's what we need, truly. We need that in order to help drive and fuel our research. I also do want to just note that there's been quite a lot of comments as well, Dory, just saying that this is wonderful news, that they love your webinar. And I agree, you have made this extremely easy to follow. Some of those graphs, I was personally a little bit, oh goodness, what is this? But you were able to explain and simplify what they were showing us in such a fantastic way that just made it digestible for all of us. So thank you so much, Dory, for taking your time. I mean, being a PhD candidate, I'm sure your time is limited. So we appreciate your efforts in being here. And echoing what you said as well, Dory, thank you to all of our participants who are on the call this evening. Whether you are a patient with myeloma or someone who's loves someone and supporting someone with myeloma, it's really great that you are here and taking an active role and learning about things that are happening on your journey, because knowledge is power. That's what I believe.
I can just
Thank you
add
so
to
much.
this, Chantal, actually. So we do understand research takes time. Of course, we're all excited. We want to have it tomorrow available for people. Um, I'm sure there's a lot of data, a lot of work behind this. So thank you so much for your involvement. To those people online, just want to mention so what Dory showed us about proteins and translocation. So if you want to have more information regarding this and better understand, we do have at myeloma.ca in the resource library an info guide which is called Understanding Your Blood Test. So if you want to learn more about your own case, probably you had information from your doctor, you're not sure. So you can refer to that tool too. So thank you so much, Jerry. I think we're going to end the session there and invite you over probably in some months or years from now with other topics. Very interesting to have you on board, and thank you for your involvement.
Thank you so much. I really appreciate your time and for having me. And for the audience, thank you for listening and your questions.
Thank you, Dory.
Thank you. Good night, everybody.